Okay, Dr. Kastner, we're about 5, 4, 3, 2, 1.
Okay, to all of you around the world who are watching this 8th program in the 21st year of Demystifying Medicine at the NIH, I say good morning, good afternoon, or good evening, whichever is appropriate. We're delighted that you are joining us. The goal of this program is to bridge the exciting developments in biology and engineering with medicine. The sessions are on Tuesday afternoons from 4 to 6 Eastern Standard Time from January 4th to May 3rd. You can access this through the NIH video cast. The indication is on the slide. The entire course schedule is also available, and we are
recording in progress.
For those who attend at least 50% of the sessions and pass a final exam, which is a multiple choice and not a stress test, you can receive a certificate. Physicians who seek a CME credit, please note that the code for today's session is 38648. Now, all those attending today's program, you can submit questions during the presentations by sending it through the Send Live Feedback on the video cast display on your computer. Please note that all previous sessions for the past 20 years, are available on the NIH Videocast network, which is indicated on this slide. If you have additional information, please feel free to contact me, and my email address is also listed. May we have the next slide, please? Now, the logo for this course is what many of us think is the most famous bridge in the history of the world. It is also, by the way, the first question on the final exam that will be given to you. It's the Brooklyn Bridge, connecting Manhattan and Brooklyn. And it is symbolic of the bridging between advances in fundamental science and medicine. And like the physical bridge, there are disagreements on both sides of a gap as to whether a bridge should be built, and if so, how. And most importantly, once a bridge is built, life is never the same on either side. So we hope that these programs, which link all aspects of biology, engineering, occasionally space, or the deep sea vents of the ocean, and next week, the origin of life, will be stimulating and provoke those who attend and listen to think more deeply. The intent of these sessions is not to overwhelm you with yesterday's research data or a massive amount of information to be regurgitated on some test. Now, we're very fortunate, next slide, please, because today's program is the first in which we have 5 participants, and it deals with the taxonomy of autoinflammatory disease. You all know that the ancients recognized inflammation by virtue of a combination of pain, redness, loss of function, and heat or fever. And in most instances, that fever was due to some form of infectious agent. But with time, it turns out that inflammation has many causes other than infection per se. And one of the more striking groups of inflammation, inflammatory diseases, are those that rely upon inheritable defects in the basic process that leads to inflammation, autoinflammation. autoinflammatory disease. And our speakers today, and there will be 3 plus 2 patients, the first time we have ever had a quintet in the 20 years of this course, they will be discussing under the title of Reimagining the taxonomy, the classification of autoinflammatory diseases. Now, the ringleader in this program is Dr. Dan Kastner, who graduated in philosophy from Princeton, received MD, PhD from Baylor, followed by a medical residency, came to the NIH in 1985 and has been here ever since. He did a prolonged stint as the scientific director of the National Human Genome Research Institute, from which he is now emeritus, and is currently an NIH distinguished investigator and chief of the inflammatory disease section. He is also, notably, the co-chair of the course in Demystifying Medicine. Dan's research has been of extraordinary excitement and importance. He became interested in inflammatory disease, fever, for which there was no known infection. And you probably know that one of the more common chief complaints of patients admitted to a hospital is fever of unknown origin. Within that large group of etiologic factors, there are patients who have genetic disturbances that lead to fever, and they lead to inflammation too. And it was through the study when he came to NIH of familial Mediterranean fever, for which he identified the fundamental defect and discovered the agent, pyrin, and later participated in cloning the gene that opened the door to autoinflammatory diseases as disorders of the innate immune system. And he has pursued throughout his brilliant career at NIH the elucidation of autoinflammatory diseases, their mechanisms, their genetics, and means of therapy. For this work, Dan has received many awards and honors, including election to the National Academy of Sciences, the National Academy of Medicine. In 2018, he was the Federal Employee of the Year, and more recently received the Ross Prize in Molecular Medicine and the Crawford Prize in Polyarthritis. Next slide, please. Now, Dan will be joined by 2 professional colleagues. Dr. Daniela Schwartz of the National Institute of Allergy and Infectious Diseases graduated from Rice, an MD from Wake Forest, medical residency at Virginia Commonwealth University, trained in clinical rheumatology at the NIH, followed by a Metzger Scholar Fellowship with John O'Shea at NIH, and in 2018 became an assistant clinical investigator in the NIAID Intramural Research Program. Notably, Daniela was recipient of the prestigious American Society of Clinical Investigation Young Physician Scientist Award in 2020. And she will be joined by Dr. Kalpana Manthiram, an assistant clinical investigator in NIAID as well. Kalpana received her medical degree from the University of Texas Southwestern, a residency in pediatrics at Boston Children's and Boston Medical Center, a pediatric infectious disease fellowship at Vanderbilt, and it was there that she began to work on the most common periodic fever syndrome in children. She then was a postdoctoral researcher in the Kastner lab in the Genome Institute, where she has studied the genetic risk loci and pathogenesis of periodic fever syndromes. She's been very interested in the immunologic mechanism of mucosal inflammatory diseases to develop treatment guidelines, and she provides medical care for patients with autoinflammatory diseases. So, with this introduction, we all are anxiously awaiting the exciting presentation to follow. And so, Dan, please carry on.
Well, thank you so much, Wynn, for that kind introduction. I hope that you all can see my screen, and let me just put it into presenter view and turn on the laser pointer. So anyway, Wynn, that was just a wonderful, very kind introduction, and it is my enormous honor and pleasure to be here this afternoon, or this morning, or this evening, or whenever it is for those of you who are participating in this event. So in any case, we are going to be talking about the autoinflammatory diseases and thinking about how they are organized. And probably before we go even a little bit further, there's a little bit of housekeeping that we have to do. I should disclose to you that, in fact, I and my colleagues, Dr. Schwartz and Dr. Manthiram, have no relevant financial conflicts to disclose, and our learning objectives. At the end of this session, the participant should be able to explain the difference between autoimmune and autoinflammatory diseases, recognize the salient clinical features of selected autoinflammatory diseases, and explain how the tools of human genomics have advanced our understanding of the autoinflammatory diseases. And in the spirit of these sessions, as Dr. Arias pointed out, we are all about building bridges. And here you see a photograph of my lab taken before the pandemic, so social distancing was not even imagined at that time, and no one was wearing a mask. My lab, and then my clinical team also before the pandemic. And of course, the bridge that brings them together is our patients. And of course, that is what really inspires us in terms of posing new questions to us, and that drives us to try to understand human disease and to develop targeted therapies. Of course, I should also mention, for those of you who are not immunologists, just a little bit about the two flavors of immunity. First of all, on the left, we have adaptive immunity, the form of immunity that many of you think of when we talk about the immune system, in which T and B lymphocytes are the major players, in which the genes for receptors somatically rearrange and mutate. Think about immunoglobulin genes or T cell receptor genes. And the autoimmune diseases are characterized by high titer autoantibodies or antigen-specific T cells. On the other hand, we have the phylogenetically more ancient innate immune system, in which myeloid lineage cells, such as granulocytes or monocytes and macrophages, are the major effectors, where the receptors or the genes for the receptors are hardwired in the germline genome, and in which the autoinflammatory diseases are characterized by seemingly unprovoked inflammation, but without the high titer autoantibodies or antigen-specific T cells that we would see in the autoimmune diseases. Now, in fact, my introduction to the autoinflammatory diseases occurred before the term autoinflammatory even existed. And that happened on the 14th of November of 1985, when I, as a new rheumatology fellow at the NIH, saw a patient, a young man of Armenian ancestry, who presented to us with a history of lifelong attacks of fever, usually on the order of once a month, with arthritis, affecting either one joint or sometimes a couple of joints at a time. Usually it would be the knee or the ankle, and the knee could swell up to an enormous size, you know, like a grapefruit, as they say. After hearing his story, I didn't know what really he had, but fortunately there were colleagues of mine in the lab from Israel who did recognize this story very well and pointed out to me that this was probably familial Mediterranean fever. So for those of you who are not familiar with FMF, let me just digress for a moment to say that FMF was first described, at least as a series of patients, by Dr. Shepard Siegel, a physician at Mount Sinai Hospital in New York in 1945. And at that time, he described 10 patients who were of Jewish ancestry and had a history of recurrent episodes of severe abdominal pain, sometimes prompting exploratory laparotomy. And he called that disease, because there was no infection associated with it and it was not cancer, he called it benign paroxysmal peritonitis. Now, that disease was then recognized in Israel as Jewish populations were emigrating to Israel in the 1950s, and it was dubbed familial Mediterranean fever at that time. It was determined by segregation analysis that it was a recessively inherited disease, and other groups found familial Mediterranean fever common in Jewish, Arab, Armenian, and Turkish populations, as well as some other Mediterranean populations. The clinical features of FMF include, as the name says, fever, also sterile peritonitis, pleurisy, inflammation of the lining around the lungs, arthritis, as it was the case for my first patient. And then some patients will have the deposition of an inflammatory protein, of a cleavage product of an inflammatory protein in the kidneys and other vital organs. And before there were treatments for FMF, those patients could develop kidney failure and even die from the kidney failure. So back when I first saw that patient, this was incredibly exciting as an interesting disease. The cause of it was not known. It was very stimulating to think about, well, what could be the cause of it? And certainly at that time, the Human Genome Project was just coming to the fore as a way maybe of figuring out what the cause of FMF might be. And I sort of took to this in earnest once I'd finished my clinical work as a fellow, which was around in 1988. And so the question was, in 1988, could we find the recessive gene that is mutated in familial Mediterranean fever? And just as a little bit of background on that, in 1978, 10 years before, Dr. Y.W. Kahn, who was and still is a professor of hematology at University of California, San Francisco, for the first time ever found that a polymorphism in DNA, a variation in DNA, could be identified and co-segregated with disease in a family with sickle cell anemia. The first time that a DNA marker that segregated with disease in a family had been identified, 1978. And then a couple of years later, in 1980, David Botstein said, well, if you can do that with one marker in one family, then we could make a whole bunch of markers and from a whole bunch of families, and in fact, identify a map, build a map of markers throughout the human genome, and could use that to map human disease genes and even to identify those genes eventually, 1980. And then in 1988, here's Francis Collins, hot in the pursuit of the gene for cystic fibrosis, although he had not yet found it in 1988. So at that point, it did seem like, well, maybe we could find the gene for familial Mediterranean fever. It hadn't yet been done in exactly that way, that far, but maybe worth a try. And so I established a collaboration with the Sheba Medical Center in Israel and had a field trip in the summer of 1989 to Israel, where with my colleague and collaborator, Professor Mordechai Pras, we visited a number of families with familial Mediterranean fever in Israel. Some of them came to his clinic, some of them we went to their houses. Here is a photograph of one of those families. This photograph has been adjusted to comply with HIPAA requirements so that no confidentiality would be breached. But in any case, here they are, one of those families, and here's a whole set of pedigrees that we drew blood from during that time. And here's my lab a year or so later. Ivona Aksentievich, my first postdoc, who's gone on to have a very distinguished career in human genetics, Louis Gruberg, who went on to become a cardiologist. And there we were trying to use the new techniques of human genomics to find that gene. But at that time, there were only about 300 markers throughout the whole human genome. So there was at least a chance that we'd never find it if the gene was in the wrong place relative to those markers. Well, fortunately, we persisted, and eventually we did figure out that the gene for FMF, this was in 1992, is on the short arm of chromosome 16. So that was great. But then we had our next job cut out for us, which was actually to develop maps of that region of chromosome 16, because it had not been mapped. And so it was up to us as the explorers of that region of chromosome 16. And we had a couple of genetic markers that ultimately we had narrowed things down to. And then we used a set of cosmids. Cosmids are chunks of DNA, about 40 kilobases in length, that had been developed actually at a laboratory, a Department of Energy laboratory at Los Alamos, the home of the atomic bomb. So I had gone out there and set up a collaboration with them, and we established this map going from telomere across to the centromere. And ultimately, we were able to create these overlapping cosmids, a map of these overlapping cosmids that gave us this region, which was about 1 million base pairs in size and covered by 170 or so cosmids. Using that, we were eventually able to narrow things down to about 100,000 base pairs, 100 kb. And then we used a type of plasmid that allowed us to pull out exons, because you see these cosmids had not been sequenced at that point, to pull out exons. And here's the exon that was the toehold for us to find actually the gene for FMF. And that took us until 1997. So 9 years altogether from the time of starting this project to actually finding the gene mutated in familial Mediterranean fever. And we denoted that protein as pyrin after pyrexia for fever. Was it worth it? Absolutely, it was worth it, because this turned out to be a gene that encoded a protein that was not known before at that time. That was the beauty of human genetics and genomics, especially at that time, is that you could find these things that were totally new and hadn't been seen before, recognized before, imagined before even. And of course, I had a number of collaborators. We had a number of collaborators at that time. Right off, we found some really interesting things about the population biology of FMF. We found that modern day carriers of FMF mutations, in fact, are descended from ancient founders from biblical times, probably. So that, for example, there's this one mutation called V726A, and the path, the projected path of V726A is shown on this map. So V726A is found in the Ashkenazi Jewish population of Eastern Europe. It's found in the Turkish population, Armenians, Iraqi Jews, and even the Druze, an isolated population in the Middle East. And how do we know that they're all connected with one another? Because they have the same mutation on the same DNA fingerprint of chromosomes. And so basically we can tell by the other DNA markers that in fact there was a common ancestor. Similarly, for the M694V mutation, you see the dotted line here, a dashed line. That mutation is seen in the Sephardi Jewish population of North Africa, as well as the Turkish population, the Armenian population, the Iraqi Jewish population as well. Again, same mutation on the same ancestral chromosome, a different ancestral chromosome from V726A. So we'll come back to that in a little bit, but that was a fascinating observation. In addition, we did learn something about the biology of inflammation. We learned eventually, not right away, it took a little bit of time, but we learned that in fact the N-terminus of this protein, this pyrin protein that's encoded by the gene mutated in FMF, encodes a domain which is called these days the pyrin domain. And this pyrin domain forms a 6-alpha helical structure, which allows for the formation of a charged dipole. And so the charged dipole, in fact, you can see it here, the positive charges are in blue, the negative charges are in red. And so you can have a situation where the pyrin domain of one protein interacts with the pyrin domain of another protein by virtue of positive negative charge attractions. And so this actually turns out to be a fundamental way that proteins of the innate immune system can interact with one another, is by cognate like pyrin domain interactions. And there are some 20 proteins in the human genome that have a pyrin domain. Now, to illustrate to you what actually happens, we have a silent movie. So here we go. This movie is entitled The Inflammasome. And the inflammasome is a macromolecular complex that activates certain cytokines, certain mediators of inflammation. And here we see the star of the show, the inactive NLRP3. It is a protein. It's not pyrin. It's kind of like pyrin. It has a pyrin domain at the end. The pyrin domain at the end terminus is that red clown nose appearance thing there, right on the end of the NLRP3 protein. And the NLRP3 protein can form an inflammasome, something that activates cytokines. Pyrin can do that too. So here, it's just that we didn't make the movie. The NLRP3 people made their movie. So in any case, here you see this disc forming. And it's forming by the interaction of pyrin domains, like interactions of pyrin domains from different proteins. And here it is coming on a side view. It looks like a flying saucer coming in. And now you're going to see something really incredible happen. And that is that you have these other molecules that are coming in that are called ASC. I won't tell you what ASC stands for, but they also have a red pyrin domain at one end of them. And you see it's forming a filament of like interactions of pyrin domains, and then sticking out on the outside are these brown domains, which are called card domains, which are similar in structure to pyrin domains, but they have their own set of interactions. And so now you're going to see caspase-1, which is an enzyme that can cleave cytokines. It's coming in and binding to these filaments by virtue of the card domain interaction, because you see caspase-1 has a card domain and ASC has a card domain. And now they're assembled there, they're self-activating, and now they're cleaving molecules of IL-1 and IL-18, which are pro-inflammatory cytokines. And now here you see coming in gasdermin D, that blue and gray molecule. Gasdermin D is one of the most toxic things known to man, woman, or child. The N-terminal fragments of gasdermin D are floating off to the right now. The inflammasome is disassembling and floating off. And in a moment, we are going to see the N-terminal fragments of gasdermin D coming in for the final action. So here we see the gasdermin D molecules. You see the gray gasdermin D N-terminal fragments, and they're going to come and they're going to form a circle. And that circle is going to form a pore that can insert itself into the cell membrane, disrupt the osmotic gradient across the cell membrane, and cause the cell to burst. And when the cell bursts, there it goes, then the cytokines that were activated by the inflammasome, IL-1 and IL-18, get released from the cell, and a bunch of other things that trigger inflammation also get released. So that is the story of how this all works. And with regard to pyrin, pyrin can form an inflammasome too. It is regulated by this molecule up here called Rho-A, which is something that lives on the inside of the cell membrane. Ordinarily, Rho-A activates a process by which pyrin is phosphorylated. And when it's phosphorylated, there's an inhibitory molecule that binds to pyrin that prevents the formation of a pyrin inflammasome. When Rho-A is disrupted, that process is stopped, and then you can form a pyrin inflammasome. What causes Rho-A to be disrupted? Well, actually, bacterial toxins disrupt row A. So in fact, the pyrin inflammasome is formed when a cell gets infected by certain bacteria. So it's a defense against those bacteria. Now, one can also ask, or at least someone might ask, I was asking, we were asking, our group was asking, and I think you would want to know too, why is it that there are such high carrier frequencies for familial Mediterranean fever in multiple Middle Eastern populations? Well, you probably didn't know that there were such high carrier frequencies, but there are. And the total carrier frequencies for Fmf mutations in the Turkish, Armenian, Arab, and non-Ashkenazi Jewish populations type allele. It's like a spike there. It does not have a wide haplotype. That's the kind of thing you want to see if you're looking for evidence of selection. You can also say that that haplotype is on the tail of the curve of distribution of similar haplotypes, haplotypes of similar frequency in the Turkish population. It should be at the tail of the curve if there's selection going on. From that, you can actually then, by mathematical approaches, you can calculate a coefficient of selection for Fmf mutations in the Turkish population, and that turns out to be 0.077. Put that in perspective, well, the selection coefficient for lactase persistence, the ability to digest cow's milk in the Northern European population, which is widely cited as an example of something under selection, is 0.056. So quite high selection coefficient. And that even allows us to estimate the age of these Fmf mutations. It ranges somewhere between 2,000 and 5,000 years. So probably it did arise in biblical times. So then, of course, you can ask the question, well, what would have been selecting for these high Fmf carrier frequencies? And that's illustrated on this slide. We, of course, were thinking about the usual suspects, and certainly Yersinia pestis would be a usual suspect. And it turns out that the plague bacterium does make toxins which can activate the pyrin inflammasome, YAP-E and YAP-T. So the pyrin inflammasome, in fact, should be a defense against plague. But in addition to that, plague makes a toxin which inactivates, poisons the pyrin inflammasome. But in people who have FMF, the pyrin is immune to that inactivating process. And so here on this slide, it just shows that in normal people who have a normal version of the FMF gene, that FMF protein pyrin gets heavily phosphorylated by the plague YAP-M toxin. But people with FMF, their pyrin is resistant to that effect. So they do not have that same effect of plague toxin. That results in increased IL-1 production, both by carriers of the FMF mutation and people that have FMF. We've made knock-in mice that have these mutations, and they're, at least to some extent, protected from infection by Yersinia pestis. And one can even simulate the frequency of FMF mutations, for example, in the Turkish population, using the information that we've gotten in terms of selection of coefficient, age of the mutation, and some historical data. And the carrier frequencies are pretty much what one would expect based on what we have seen in that population. So in any event, that's just a little bit, maybe more than what you wanted to know, but it's interesting, I think, anyway, information on FMF. While all that was going on, we were seeing other patients that have unexplained recurrent fevers. And it turned out that a group of those patients actually had a different kind of periodic fever where the attacks lasted much longer than in FMF, where it was dominantly inherited rather than recessively inherited, and where the patients didn't respond to the usual treatment for FMF. And in 1999, a couple of years after we had found the FMF gene, we found that at least some of those patients have mutations in the TNF receptor gene. And we called that disease TRAPS, the TNF receptor associated periodic syndrome. And we came up with this term autoinflammatory syndromes at that time, because in both cases, these are diseases in which patients have recurrent fevers, seemingly unprovoked, without the high titer autoantibodies or antigen-specific T cells of autoimmune disease. So we didn't know whether there were going to be any more autoinflammatory diseases or not, but we let the imagination run wild, and we wrote this paper in 2000 suggesting what other diseases might be autoinflammatory diseases and even proposing a classification scheme for them. And one group of them we suggested was the familial urticarial syndromes where people would have fevers and break out with hives when they had fevers. There are such diseases as that. And then about a year later, much to our surprise and delight, Dr. Hal Hoffman at the University of California, San Diego, published a paper in which he showed that those two diseases, the familial urticarial syndromes, in fact, the gene mutated in those diseases encodes a protein with a pyrin domain. And that protein actually is NLRP3, which of course then led us, because at that point, NLRP3 and IL-1 activation that was beginning to be understood, led us to think that perhaps we could treat patients with the most severe form of disease of that sort. We could treat them with IL-1 inhibitors. And this is work of my colleague, Raffaella Goldbach-Mansky, and I from back published in 2006 on a disease called NOMID, neonatal onset multisystem inflammatory disease. This is the before and after. Before treatment, these patients had hives, they had conjunctivitis, they had inflammation of the lining around the brain, which could lead to blindness and deafness and intellectual disability. They had inflammation of the cochlea that could lead to deafness. After a course of anakinra, the IL-1 receptor antagonist, a potent IL-1 inhibitor, this is what we saw. The rash went away, the conjunctivitis went away, the aseptic meningitis went away, the cochleitis went away. This work and other work, in fact, was the foundation for FDA approval of this drug, this biologic, in NOMID. And in fact, the kids that have been treated with this are still on anakinra 20 years later. And instead of having a very poor prognosis, we know of a number of them that are now attending college. So it's really a wonderful thing to see. So now we're on to the next chapters and to my colleagues. So the next chapters are written because the cost of DNA sequencing went down dramatically after about 2007. And so this has led to an explosion in terms of the number of autoinflammatory diseases that we can identify, in which pathways other than IL-1 are involved, in which there are many monogenic diseases, and in which, in fact, there are genetically complex disorders as well. This is just a figure from a paper that was published in Science a few months ago on the autoinflammatory diseases, just highlighting 10 of the diseases. There are now around 50 monogenic autoinflammatory diseases, and lots of room for changing the taxonomy, improving the taxonomy of these diseases. And we're going to focus for the rest of this session on disorders in which patients present with fever, but also with painful oral ulcers, painful sores in their mouths. One such disease is a disease called Behçet's disease, which is also seen in the Middle East, as well as along Marco Polo's Silk Route going to the Far East, to China and Japan and Korea. And patients with Behçet's disease have painful oral ulcers. They can have inflammation of the eye. This is pus in the anterior chamber of the eye. These patients can have ulcerations in the genital region. And they can also develop something called pathergy, where they will, if their skin is pricked with a sterile needle, they may develop a pustule in that site. So in any case, a fascinating group of diseases, and I'm sure that you will be delighted as Doctors Schwartz and Manthiram regale you with their adventures studying these diseases. And so at this point, I will turn things over to Dr. Danny Schwartz.
Hello all, can you see my screen?
Yes.
Okay, I'm going to put myself on laser pointer, and I want to start by thanking you for the opportunity to share this research with you. I will be talking to you about a disease that is very near and dear to my heart, haploinsufficiency of A20. And after Dr. Kastner has told you all about autoinflammatory diseases that really in isolation involve the innate immune system, I'm going to tell you about situations where autoinflammation isn't just autoinflammation, using Ha20 as a sort of cardinal example. As a disclosure, I just want to let you know I will discuss the general concept of targeted off-label therapies for this disease using a various number of targeted treatments that we've used for these patients that are off-label. I would like to start by telling you about one of the index patients for this disease, and a patient whom you will be hearing from directly at the end of this talk. And this young lady was diagnosed at the age of 10 with juvenile idiopathic arthritis. And this is because she developed very severe arthritis with systemic inflammation. She had some response to standard biologics that you use to treat this disease, but not a complete response. And you can see here, these aren't her joints. This is another Ha20, but this is a very typical example of what she had. As time went by, at the age of 14, her disease worsened despite treatment with standard biologics for juvenile idiopathic arthritis, and she began to develop oral ulcers, after which she developed very severe neurologic disease, including uncontrollable movements and very severe retinal inflammation that led to blindness with a very rapid onset. Because of this, she was diagnosed with systemic lupus erythematosus, and she was treated very, very aggressively with lupus-directed therapy, but unfortunately had an incomplete response and continued to suffer from both the neurologic disease and continued joint disease and oral ulcers. And at this point, at the age of 15, was referred to the National Institutes of Health lupus protocol for an experimental autologous bone marrow transplant, where she received her own bone marrow and a very intensive conditioning and immunosuppressive regimen. On that treatment, her symptoms transiently got better for about a few years, but then between the ages of about 22 to 23, they came back, and they came back with a vengeance. She had worsening neurologic symptoms. She would have episodes of, again, uncontrollable movements. Her platelets began to be destroyed, and this was an autoimmune destruction called ITP. Her oral ulcers came back, her joint inflammation came back, and she began to also experience genital ulcers. And this really began to leave the domain of what you would typically see in a patient with lupus. And around this time, due to her extensive family history, genetic testing was performed by our group. And we indeed identified a mutation in this gene TNFAIP3, which encodes for a protein known as A20. And you can see here, P6 right here is our patient. This is her family. And we found that she had a biological sister, mother, and aunt who had similar symptoms to her. And this mutation segregated with disease. Around the same time, we were following another family with Behçet's, and we found mutations in the same gene in that family. You can see here how we came to that gene. And then through working with various collaborators who followed other families with inherited forms of Behçet's, we also found mutations in TNF-AIP3 in 3 additional families, as well as in one other family from a larger cohort of Turkish and Japanese Behçet's patients. We then went on, and here again, you can see this is our patient, and we're able to see that compared with a healthy volunteer, the expression of A20 protein was reduced in this patient's cell. So this was a haploinsufficiency, not enough of the protein. She did not express the mutant form, so really it was just that she didn't have enough of this protein. Which then brings us to, how did this lack of A20 come to cause disease? Well, A20 prior, so in its very first days, is best studied as an inhibitor of NF-kappa B signaling. So Dr. Kastner told you about mutations in TNF receptor. When TNF comes and activates its receptor in induction of inflammation, this activation leads to ubiquitination, so the tagging on of ubiquitin molecules into various positions of downstream signaling molecules. And some of these modifications activate and some of them inactivate. 2 of the activating modifications are the sticking on of ubiquitin to lysine 63, as well as linear ubiquitination. And A20 removes K63 and linear ubiquitin from signaling molecules like RIP1. And so because these modifications activate, the removal inactivates these signaling molecules. Additionally, ubiquitins can be tagged onto position lysine 48, and that targets molecules for degradation. And A20, in addition to removing these ubiquitins, also will add ubiquitins onto position lysine 48 and targets those same molecules to degradation. So 3 mechanisms by which A20 acts as a modifying enzyme and targets this inflammatory pathway for inactivation. As you can imagine, not enough A20, too much NF-kappa B signaling, and increased inflammation. And indeed, when you look at cells from our patient with HA20, that is what you see. So here you see increased I-kappa B alpha phosphorylation in our patient, increased I-kappa B alpha degradation, and increased phospho-IKK alpha beta. And so that's increased NF-kappa B signaling, very consistent with enhanced inflammation and reduced A20 in our patient. But one inflammatory pathway isn't enough. A20 also inhibits the NLRP3 inflammasome, which Dr. Kastner told you all about in the context of NOMID. And so work by Avril Ma's lab has demonstrated this. And indeed, when you look back at our patient, you see that she also has increased IL-1 beta relative to a healthy volunteer, and that this does go through the inflammasome. You treat our cells with an inflammasome inhibitor, and they no longer overproduce IL-1 beta. But apparently, 2 inflammatory pathways still aren't enough. A20 is also important for inhibiting this inflammatory form of cell death known as necroptosis. And defects in necroptosis can also lead to autoinflammation in a syndrome called CREA. So work again from Dr. Ma's lab has shown that in an A20 deficient cell, you will see enhanced necroptosis or inflammatory cell death. And there is yet a 4th inflammatory pathway that A20 seems to repress, and that is type 1 interferon signaling, which has been described in the pathogenesis of other autoinflammatory diseases by Dr. Goldbach-Mansky's group, such as Candle and Sabi. And we have seen that patients with haploinsufficiency of A20 do indeed have increased expression of interferon-stimulated genes and increased phosphorylation of STAT1, which is activated downstream of type 1 interferons. And so now we have a situation where rather than having one dysregulated pathway, we have at least 4 potential inflammatory pathways that are dysregulated. And to make matters even more complicated, I told you that A20 has multiple functions that are involved in ubiquitin editing, and each domain of the protein is important for a different ubiquitin editing function. So for example, this zinc finger 7 domain is very important for linear ubiquitination and deubiquitination. And this domain here, the OTU domain, is important for taking off K63 ubiquitins. That's part of the activation that I told you about of NF-kappa B, whereas this area here, zinc finger 4, is important for K48 ubiquitination and degradation. And very recent work by Dr. Ma's group has shown that this domain here is very important for preventing arthritis and TNF-dependent responses, whereas you really need both zinc finger 4 and zinc finger 7 together in order to suppress the inflammasome and IL-1 beta production. And so now coming back to our syndrome, haploinsufficiency of A20, we have multiple different domains of A20 that inhibit different inflammatory pathways, 4 inflammatory pathways. And coming back to our patients, we have to ask, could any of this cause different phenotypes in patients with haploinsufficiency of A20, which was originally described as a Behçet's-like disease? And so in order to answer that question, I'm going to take you back to something that Dr. Kastner told us at the very beginning of his talk, which is that autoinflammation is caused by a dysregulated or overactive innate immune response without autoimmunity, without high titer autoantibodies or a dysregulated adaptive antigen-specific response. And this is a very classic view of the disorders of immune dysregulation, where each one has its own bin. But this is the view in 2022. So as sequencing has become cheaper and cheaper, and we've discovered more of these diseases, we can see that there are genes that are expressed in innate cells and in adaptive cells, and that are important for innate immunity and adaptive immunity. And no, I think that no disease is more emblematic of that than HA20, because TNF-AIP3 is expressed everywhere and is important for many different types of biologic processes. And so, for example, A20 mutations do cause autoimmunity. If you look at patients with haploinsufficiency of A20, many of them do have bona fide high titer autoantibodies that you don't see in diseases like FMF or TRAPS. You see autoimmune thyroid disease. We have patients with kidney disease that looks indistinguishable from lupus nephritis. And these are very clear autoimmune manifestations. And autoimmunity is not the only type of immune dysregulation we see. We also sometimes will see patients with immune deficiency. And in some patients, this manifests with recurrent respiratory infections. But we've also seen patients who present with recurrent gastrointestinal infections, or even diseases that mimic severe combined immunodeficiency. And then sometimes we'll see patients with yet another type of immune dysregulation. A20 is also important for cellular proliferation, and somatic mutations in A20 are even linked to lymphoma. And so haploinsufficiency of A20 has been described to cause lymphoproliferative inherited diseases in certain patients. So yet another type of immune dysregulation. And then finally, just last year, we did a population survey of multiple autoinflammatory diseases and found a signature for both clinical and immunophenotypic findings consistent with allergy in patients with haploinsufficiency of A20. And A20 is actually even linked to the development of common asthma. And so now we have a disease that crosses multiple different types of immune dysregulation and really does expand our conception of what it means to have an autoinflammatory disease. And then the question becomes, this isn't something like a pyrine mutation where you have one pathway and you can target that pathway, or in the case of NOMID, again, you target IL-1 beta and you treat the disease. We have multiple dysregulated immune pathways. How can we treat these patients? And so we're left with trying to target some of the cytokines and pathways that A20 is important for repressing. So for example, we can use inhibitors of TNF because A20 represses activation of pathways downstream of TNF, or we can target IL-1 beta because we know that A20 is important for repressing the inflammasome, or we can use clinical JAK-STAT inhibitors to target responses downstream of type 1 interferon. And we have done all of these things in various patients with some success. And so coming back to our patient, how did we target her disease? Well, I told you that at the age of 23, we diagnosed our patient with haploinsufficiency of A20. And we saw that she had increased production of IL-1 beta. And so we knew that inflammasome targeted treatment was a successful way to address many of these other autoinflammatory diseases. We knew that the inflammasome was activated in her case. And so we treated her with inflammasome targeted treatment. And this was incredibly successful for about 6 years. She was nearly in remission. She had very low disease activity with some flares that could be addressed with an increased dose. And she was very successfully treated until about age 29. And at that age, she began to develop increased back pain, which then progressed to increased arthritis, increased systemic inflammation, and then finally neurologic symptoms. And this led to an escalation of her therapy until we were suppressing multiple A20 associated pathways, including the inflammasome and including JAK-STAT signaling and systemic steroids. And she continued to have symptomatic flares with neurologic symptoms and neuroinflammation despite very, very, very intensive immune suppression with many different pathways being targeted. And the question then became, did we have anything else to offer this patient or a patient like her who is on multiple targeted therapies or even broad therapies and still uncontrolled with potentially organ-threatening disease? And the answer is that yes, there is. This is very, very new data, but in 2 patients now, there have been reports that an allogeneic stem cell transplant, now this is different from the transplant our patient had, which had been autologous with her own marrow. This is essentially giving a patient somebody else's immune system. And so these are 2 pediatric patients who are on multiple therapies that were not successful and were treated with allogeneic stem cell transplants and were able to get off of their medications and had very successful outcomes. They did have some damage from their prior disease, but they were successful both in terms of their engraftment and their response to transplant. And that is indeed the course that we chose to follow with our patient. She did elect to pursue this transplant, which we hope will be, which will start within the next couple of weeks. She actually was admitted just this week, which is why she couldn't be here in person today, and why her interview had to be recorded. Before I proceed onward to her interview, I want to first acknowledge and thank all the people who contributed to this whirlwind of research of HA20 that I've put you through. And that includes students and members of my group, and most particularly Sarah Blackstone, who did a lot of the assays with the interferon production in A20, as well as my mentors at NIAID, Josh and Pam, of course, the big group at NHGRI that were instrumental clinical mentors during my fellowship, and all of the HA20 patients and their families who generously agreed to participate in the research. And then I'm going to allow us to proceed to our recorded interview with this patient who generously agreed to share her thoughts despite preparing for her transplant. I think that I need IT to proceed to the interview. Sorry, guys, I think we're having slight technical issues with the loading of the patient interview.
Hi there, it'll be just a moment. Please hang with us.
So
Thank
much
you.
for joining us today and for sharing with us your experiences living with HA20. I'm going to ask you some questions both about living with HA20 itself, as well as being a patient with a complex inflammatory disease more generally. And I'm going to ask, I'm going to start by asking you if you could please describe to us the symptoms that you and your family members have had over the last several years, and specifically how they might be similar or different between you and your family members.
Thanks for having me today. So the interesting thing about my family and I is that the way in which our disease manifests with symptoms really vary in scope and severity. We've all experienced at one point or another skin inflammation, joint inflammation, and the oral and genital ulcers. In addition to these symptoms, one of my family members also experiences the GI and kidney inflammation. And then for me, I am experienced or have experienced in the past the neuroinflammation, eye inflammation, kidney and GI inflammation, and ITP.
So a lot of variability between you and your family members, as well as some common things. Looking back, we've reviewed your medical history, which was very long and complex during this talk. And I was wondering if you could tell us what symptoms you might have had that could have prompted concern for a periodic fever or autoinflammatory genetic syndrome, things like fevers or discrete flares during the course of your disease.
Sure. So, one common thing that I think most patients that live with chronic illnesses experiences is that they don't sound the alarm when they experience minor symptoms. And I definitely fall under that category. But thinking back, especially, during this flare over the past 2 years, and thinking about what the minor symptoms were in the beginning, which includes, severe head and back pain, GI inflammation, hair loss, skin inflammation, and then also, excessive sleeping and excessive fatigue.
Do you feel like your symptoms occurred more in discrete flares, or was there more of a continuous escalation of your symptoms that sort of stayed there all the time?
So prior to receiving my first transplant, my flares definitely, lasted a considerable amount of time, and they, I never really got better. They would add a new medication or change a medication around, and I would get slightly better, and then I would regress again. And it wasn't really until 2013 when I received, the right diagnosis and the right treatment option, that my disease really, was under control at that point. And I still experienced, more minor flares in disease activity, but they were very manageable with just increasing, a little bit with the prednisone or a little bit with the anakinra. And then I was able to get back to my baseline doses.
So thinking back throughout, you started to have symptoms when you were quite young and have now been dealing with this for many years. How do you feel that having a genetic inflammatory disorder has affected your life?
Sure. So, the obvious answer to this is, because I had the retina inflammation in 2005, that led to the subsequent blindness and disability. Like, this has had the most profound impact on my daily life and will continue for the rest of my life. But thinking really back to, these past 2 years and this current flare, it has been, what has been harder to deal with is the mental anguish and the uncertainty of having a chronic illness, especially in the midst of a pandemic. The fear and the panic of, having a very complex condition while also living during a pandemic has been very challenging. And it's really, one of the reasons why, this uncertainty is as to why I, decided to move towards having the transplant, because, I know that the pandemic will end, but my life will not get better. And this fear and this uncertainty and the unknown is just going to continue. And so this is my opportunity to get back to having my life back.
Yeah, and I want to acknowledge that this really has been a very difficult 2 years for you, especially the last 6 months or so as we've sort of approached this transplant. And once again, thank you for sharing that with us, because I know it's been hard having gone through it together with you. How do you feel that coming to the NIH has helped you?
So I've been coming to NIH since 2005 and had my first transplant in 2006. And I've been receiving care in many ways since then. And having access to this medical care is truly invaluable. But I think, also really important to mention is, as a patient with a chronic rare condition that is very complex, finding moments of where you're kind of a little more at peace are hard to come by. And NIH gives those moments to patients. And what I mean by that is because NIH is unmatched, in my opinion, to any other care that you can receive anywhere else, from medical staff, doctors, nurses, and staff across all departments. There's never a moment where I think my care or treatment is going to be compromised, or, I won't be given the best options. And so because I don't have to worry as much about that, I get these little moments of peace. And that's, for patients that are going through incredible health challenges, that's truly a gift.
Yeah, thank you. And I'm really glad to hear that we've been able to help you like that. And then the last thing I want to ask you is what you would like the medical community and sort of the biomedical research community to know, both about HA20 and then more generally about living with a complex genetic inflammatory disease.
Sure. So, when I think about the entirety of my, medical journey, one thing that has remained true and constant is having trust in your medical team and your doctors. And that's something that I have certainly found at NIH. And trust can be really found in many ways. And for me, what that really means is that, having this rare genetic condition means that my medical team is going to think outside the box and outside the scope of what is the general thinking. And then, kind of translating that to me, making decisions that not only will impact my life today, but also thinking about how it's going to impact my future. And so trust is really crucial to me as a patient with a chronic condition.
Thank you very much for sharing with us all of that. I know this was difficult, and particularly in this time as you've been sort of approaching the transplant. So we really appreciate it. And thank you once again. And now I'm going to go back to the talk.
Thank you.
And I think that I can't say anything more eloquent than my patient, Ms. Banty, has said about living with a chronic inflammatory disease. So with that, I'm going to turn this talk over to my colleague, Dr. Manthiram, who's going to tell us all about common variants of Behçet's disease and the genetic architecture of diseases on that spectrum.
Thank you very much, Dr. Schwartz, and thank you very much for the opportunity to speak with you today. So just pulling up my slide. Can you see my slides?
Yes.
Great. Thank you very much for the opportunity to speak with you in this Demystifying Medicine series. Now, I wanted to share with you the story of a peculiar but fairly common periodic fever syndrome called FAPA or PFAPA syndrome, when I pronounce it P silent, and how it may be a facet of Behçet's disease. I will discuss off-label use of the medications listed on this slide. So what is FAPA syndrome? So the name FAPA is an acronym that stands for periodic fever, Aphthous Stomatitis, Pharyngitis, and Adenitis syndrome. It's considered to be the most common periodic fever syndrome of childhood and was first described in 1987 at Vanderbilt University after researchers there led by Dr. Kathy Edwards saw a series of patients who had recurrent fever episodes that started in the toddler to preschool age range. And these fever episodes all seem to be the same things. They're highly stereotypical. And interestingly, these episodes had regular timing. And in some patients, the regularity of the episodes was so regular that parents could predict approximately when the next episode would occur and would start planning their vacations and major family events around that time. Typically, the episodes lasted anywhere from 3 to 5 days and happen approximately every month. So some patients have described it to me as a fever menstrual cycle. During flares, patients have, in addition to fever, aphthous ulcers, which are canker sores or ulcers on the inside of their mouth, pharyngitis or inflammation of their tonsils, and cervical adenitis are inflammation of the lymph nodes in their neck. But aside from these fever episodes, they're completely asymptomatic between their flares. They're growing and developing normally. The researchers reported that, or physicians reported that prednisolone or prednisone completely aborted the flares. Within hours, the fever would resolve. But unfortunately, in many patients, the episodes would occur more frequently with steroid treatment. And later it was found that tonsillectomy leads to complete cessation of flares in most patients with FAPA. We believe that FAPA is likely underdiagnosed, as many patients are misdiagnosed as having recurrent viral infections or recurrent tonsillitis. It really requires an observant parent and an astute clinician to see that the episodes are highly stereotypical, that there's regular timing to the flares, or that there's aphthous ulcers in some patients. So many patients go undiagnosed. In fact, some of you listening to this talk may realize that you had symptoms, something similar when you were a child. So these are some pictures of canker sores that are seen during flares in some patients. So typically these are on the inside of the lip or inside of the cheek. And in most patients with FAPA are under 1 centimeter in size and resolve in a few days, although there are some patients who have more severe ulcers. And this is an image of the exudates that might be seen on the tonsils of a patient during a flare. So long-term patients with FAPA, eventually, most patients with FAPA start having flares. This is a long-term follow-up study of patients over the course of about 20 years from Vanderbilt. You can see that with time, the episodes become shorter, ranging from initially 5 days to later close to 3 days, and the episodes become less frequent until they sort of peter out. And in this study, most patients who had resolution of their flares had flares for about 6 years. However, there were a group of patients, 9 out of 60 in this study, who had persistent fever episodes at the time of follow-up, and one patient was eventually diagnosed as having Behçet's disease. We also find that most patients with FAPA continue to have aphthous ulcers after their fever flares resolve. And we and others have seen patients in our clinic who have genital ulcers or rash, which are atypical for FAPA, but more consistent with Behçet's disease during otherwise classic FAPA flares. So already we were beginning to see clues that FAPA may be related to some other ulcerative disorders. as we continue to study FAPA, we noticed that about a quarter of patients with FAPA had a family member with the syndrome. And actually, many of these family members didn't even realize that they had FAPA. It was only after we took a very close history that we realized that their fever episodes met the criteria for the syndrome. But in addition to FAPA itself, family members of patients with the syndrome also are more likely to have recurrent tonsillitis, who have undergone tonsillectomy, and also to have had recurrent aphthous ulcers. So this graph shows the percentage of siblings of patients with FAPA who have recurrent aphthous ulcers, which is significantly higher than that of controls without the syndrome. So the familial clustering of FAPA prompted us to search for genetic risk variants. So initially, we did whole exome sequencing on familial cases, but unfortunately, we weren't able to identify rare variants that explain the disease in most of our patients. And so we hypothesized instead that FAPA is a complex genetic disorder in which many common genetic variants contribute to the risk. And in fact, many of these variants may actually be in non-coding regions where they affect the expression of nearby genes. So as we begin to shift our study of FAPA as a complex disorder, there were genomic studies of two ulcerative diseases that caught our attention. So the first is of Behçet's disease. Dr. Schwartz just gave us a wonderful presentation about a monogenic form of Behçet's disease caused by rare variants in the TNF-AIP3 gene. And these mutations have high effect size in the risk of an individual to develop Behçet's disease. In order to identify common variants, which each have a low effect size, genome-wide association studies are used, or GWASs. And in a GWAS study, thousands to millions of single nucleotide polymorphisms from across the genome are genotyped in a large number of individuals with the disease and those without the disease. And then the SNPs that are enriched or reduced among individuals with the disorder are identified, and these are the risk variants for the disease. And then when you look at the risk variants together and what their biologic function is, you can begin to understand the mechanism of various complex diseases. So for Behçet's disease, Dr. Kastner, Dr. Remmers, and their group have actually done multiple GWASs of Behçet's disease, and this has really been instrumental in helping us understand the pathogenesis of this ulcerative disorder. And they found that the most significant risk factor was at the MHC locus. In fact, the most important risk MHC type was HLA-B51, which is a class 1 MHC. But in addition to that, they found that polymorphisms in ERAP1, which is a peptidase that trims peptides to be loaded onto class 1 MHC, were also risk factors in individuals with HLA-B51. In addition, they found many variants near these genes, many of which are inflammatory genes that were risk factors for Behçet's disease. And these were involved in Th1 and Th17 activation, CD8 positive T cell activation, and myeloid cell function, in addition to maintaining the integrity of the mucosal barrier. So in aggregate, these genes have risk loci have begun to help us understand the pathophysiology of Behçet's disease. A few years later, another GWAS was published, this time for recurrent aphthous ulcers. And this was by a collaboration with 23andMe and the UK Biobank, and included a very, very large number of individuals. And they basically asked people, have you ever had canker sores? And if they checked yes, they were cases, and if not, they were controls. And they identified many risk loci for recurrent aphthous ulcers, or kind of run-of-the-mill, per se, canker sores. And interestingly, many of the exact same genetic risk variants or loci that Dr. Kastner and Dr. Remmers had identified for Behçet's disease were also risk variants for recurrent aphthous ulcers. So we see that recurrent aphthous ulcers and Behçet's disease have both phenotypic and genotypic overlap. So that now beg the question, does FAPA also have which we've found that FAPA seems to have phenotypic overlap with these two diseases. Does it also have genotypic overlap? So in order to address this, we screened 3 cohorts of Caucasian individuals with FAPA for risk variants previously associated with Behçet's disease and recurrent aphthous ulcers. And we compared those variant frequencies to those among Europeans in the general population from large databases. And we found that many of these variants were also significantly associated with FAPA. In fact, this one near the IL-12A gene had an odds ratio of over 2, which is quite high for a complex genetic disorder. The commonality of genetic susceptibility loci among these 3 disorders, we believe puts them in a common family. And in fact, we proposed that these disorders be grouped on a spectrum of disease called the Behçet spectrum disorders. And on the spectrum, Behçet's disease is on the severe end, recurrent aphthous ulcers are on the mild end, and FAPA is in between. And in clinical practice, this actually finally made a lot of sense because we see patients who lie somewhere in between. So patients who have genital ulcers or rashes during their regular fever flares. We also see patients who have severe recurrent aphthous ulcers with systemic symptoms, but don't have fever. And so this concept of the spectrum has been really important for us to understand patients that don't quite fit into these existing diagnostic entities and who sort of fall somewhere in between. It's been helpful for us to understand the mechanism of their disease and to think about what treatment modalities we may use. So in terms of the genetic risk loci for FAPA, the strongest one we identified was upstream of IL-12A, and IL-12A encodes the P35 subunit of the pro-inflammatory cytokine IL-12 and the anti-inflammatory cytokine IL-35. And in order to understand the functional significance of this variant, we obtained blood from individuals who came to the NIH blood bank, and we genotyped them for this risk allele. Those who are heterozygous are shown in red, and homozygous for the risk allele are shown in green. We extracted their monocytes and stimulated them with interferon and LPS and measured the IL-12 production. And we found that individuals who carried the variant their monocytes produced more IL-12, and also their monocytes produced more IL-23, which is another pro-inflammatory cytokine. Previously, this variant near STAT4 has been associated with increased STAT4 expression, and STAT4 is important in downstream signaling from IL-12 and IL-23. This variant near IL-10 is associated with decreased IL-10 expression. IL-10 is an anti-inflammatory cytokine. And this variant near the CCR1 and CCR3 locus is associated with decreased CCR1 expression. CCR1 is a chemokine receptor that helps monocytes traffic to sites of inflammation. So it's hypothesized that decreased CCR1 may contribute to diminished integrity of the mucosal barrier, but it may have other functions. This variant may have other functions as well. So in aggregate, these variants appear to be linked to heightened antigen presenting cell and CD4 positive T cell activation. So elevated IL-12 and downstream signaling with STAT4 polarizes and activates Th1 cells to produce interferon gamma. Elevated IL-23 and STAT4 can polarize toward a Th17 phenotype, and decreased IL-10 leads to further activation of T cells. And this fit well with what was known about the immunology of FAPA flares. So during flares, patients were found to have high expression of interferon gamma in these genes. They also had high expression of Th1 chemokines in their blood and also in the tonsil. And we've also found that they have high expression of the subunits of IL-12 in their monocytes during flares. I mentioned earlier that tonsillectomy leads to resolution of flares in many patients. So we've begun to look at the immunology of the tonsils as well. And we found that the CD4 positive T cells in the tonsils of patients with FAPA have elevated Th1 and Th17 related cytokines compared to those tonsils from controls who have anatomic issues. And we also found that the CD4 positive T cells in patients with FAPA also produce significantly more interferon gamma and IL-17 upon stimulation with PMA and ionomycin. We also found significant class 1 and class 2 HLA associations for FAPA. And interestingly, most of these were unique to FAPA in that they weren't related to the other disorders on the Behçet spectrum, except HLA-B1501, which has been previously associated with Behçet's disease and recurrent aphthous ulcers. Along the Behçet spectrum, HLA is a strong risk factor for Behçet's disease, but not as strong for recurrent aphthous ulcers, and FAPA appears to be somewhere in the middle. And as I mentioned, the HLA risk alleles are different for each of these syndromes on the Behçet spectrum. So HLA may be one factor that affects phenotype along the spectrum. So with that, it's with great pleasure that I'd like to introduce to you one of my patients, Holly. Holly's actually a physician herself, and I've had the privilege of following her and her children at the NIH for the past 6 years. Holly presented to us with an inflammatory disease that had some features of Behçet's disease, but she and her children also had FAPA when they were younger. So this was before we even realized the connection between these syndromes that we began seeing her. I hope Holly can hear me. There's Holly, thank you. So first, I want to thank you very much, Holly, for being here with us today and for sharing your story. I wanted to start by asking you to share you and your children's symptoms of FAPA and how you were diagnosed.
Yes, thanks for having me. It's an honor to be here and sharing our story. I think when everything started, our daughter was 11 months old. She's the younger of our two children, and she started having fevers, and they would be 104 or 105, and she would pretty much play through it, but wouldn't drink a whole lot. And my kids weren't in daycare, and we had a nanny in the house, and she would have these fevers, and it would be every 3.5 weeks. We could almost set an alarm to it. And I kind of laughed in your presentation. You mentioned the vacations, and we would schedule things because of Becky's fevers. And we'd go to the pediatrician and they would tell us, oh, she has ulcers and she has swollen lymph nodes. It's just a virus. And, finally I said, look, she has no ill contacts. There's something going on here. We can set an alarm to it. Nobody else is sick. And so then a workup was done, and they did some genetic testing for the familial Mediterranean fever. It was negative. And so then I did read a little bit about the PFAPA syndrome, and I thought, well, I'm going to try this prednisone. And then I did tell the pediatrician after the fact, and the fever went right away. It was miraculous. And so we continued that, but over time, the fevers, it became more frequent. Instead of it being 3 weeks, it might have been 2.5. It had the rebound effect. So Becky had her tonsils out then when she was 26 months old. And again, the fevers went away. It was great. And our son, I think he was around 8. It was like 3rd, 4th grade. He started getting fevers and sore throats as well. And I said to our pediatrician, do you think it's the same thing as our daughter had? And she said, yes, I do. So we went to the ENT and he had his tonsils out. And as all this is going on, my mother said to me, you always had fever. And I did. And I remember missing 21 days of school in 3rd grade over fevers and sore throats. And I usually would feel pretty well. My mother would just look at me and she knew I had the fever. And when we went to ENT, they said, well, we're not taking the tonsils out. It's getting warm. She's not going to get sick. And my mother kind of argued with them. And she says, she'll get a fever. It'll come in 2 weeks, I can tell you. And sure enough, it did. And I had my tonsils out and between 3rd and 4th grade. Didn't miss much school after that. And, and I think the effect of that was difficult. Like I remember missing so much school that I didn't know how to address an envelope one time. And, I remember this. And my kids Sal missed quite a bit during his process too. And, I have continued to have, my fevers went away, but I've continued to have ulcers through the years and some joint pains. And as all this was going on with my kids, I was seeing a rheumatologist because I'd have these joint things where I couldn't turn doorknobs and I was having a lot of difficulty, but nobody could ever put a finger on anything. And our son was better. And then 3 years after he had his tonsils out, he started getting, he would have these shortness of breath episodes. And I worked in the ER for 18 years, and he would come in the ER and, oh, he has pneumonia, but I never really looked at the x-rays or anything like that. Well, it turns out he had pleural effusions twice. He had myocarditis that he got admitted for, and pericarditis. And then the rheumatologist, when he was admitted, actually said, does he ever have joint pain? And I said, oh no. And then we thought about it. I mean, there were multiple times when both of my kids were having joint stuff where like Sal didn't accolate one time, and because he couldn't walk, and it was a recurrent thing. And then we were referred to the NIH, and it's been a wonderful experience.
Thank you for sharing that. I think your story really shows how it can take some time to reach the diagnosis. And I think it really requires an observant parent, as you were for your kids. So you started to mention that even after tonsillectomy, you began to have some new symptoms afterwards that were more indicative of Behçet's disease. And I was wondering if you could mention some of those and when you started to realize that there was something else going on.
Well, I've always remembered having ulcers, and I would complain about it. And sometimes it's hard to eat and hard to speak when you have an ulcer in your mouth. And my mom would say, oh, you just ate too many tomato sandwiches, and I always liked oranges. You ate too many oranges. We didn't know any different. And it seemed like there are times when I would be worse than others. Looking back on it, I think stress may have aggravated some of that. I always had some sort of a joint pain where I wouldn't be able to raise my shoulder. And we went to orthopedics, and they would tell me, oh, it's from swimming or cheerleading. In college, I couldn't walk one weekend and because my knee hurt and it swelled up, and I was crawling in my dorm room and I couldn't do anything. And I went to student health, and they told me that, oh, you have Slaughter's disease. Now looking back on it, I'm a physician. That doesn't happen to a 22-year-old. I mean, there was something more going on. And it seemed like I did have periods where I'd be okay. And then when I was a resident, I was probably 26, 27, I had some eye issues where I had trouble with my vision, and I got sent to ophthalmology, and I had several bouts of uveitis with that. And I haven't had that in a few years. And at that point, nobody could put the pieces together. And they said to me, well, maybe it's a thyroid problem. And down the road, I ended up with was diagnosed, and that's been under control as well. But I think in the last few years, my biggest problems have been the joint issues, and the ulcers have gotten worse. But right now, I am on an inhibitor and Imuran. That is really ulcers.
Thanks, Holly. I think I think as time went on, like a lot more of these Behçet's like features started coming up, as you mentioned, the significant oral ulcers, which kind of were more severe than sort of the milder ones we see in Behçet's, in PFAPA syndrome. Remember, you had also told me that you had rashes, and there were times where you had mentioned that you would develop lesions at the site of trauma to your skin.
Right.
Could you describe some of that a little bit for
us?
It
was probably like a year and a half ago. I fell and I had a scrape on my knee. No big deal. And like a week or so later, then I get like a bolus kind of lesion at that site. And every now and then, if I bump that, it does come back. I never had that with blood work, and I've had a lot of blood work, but it seems more when it's some sort of a trauma that I see that with, or if I scratch really hard sometimes and break something open, I'll see that. I also I remember having a rash where they were red wheals and this had centralized clearing. And I went to student health at that point. I probably was second year of medical school, and it was a very stressful time. And they nobody could figure it out, but eventually it went away. But I've had really strange rashes through the years. And a few years ago, I had another one, and that was biopsied, and it showed an inflammatory reaction there.
Yeah, I think your disease really exemplifies how it's hard to like pinpoint exactly what it is that you have, like a clear disease entity that it sort of defies the existing disease classifications in some ways. So I was wondering if you could talk to us a little bit about how having an inflammatory disease like this has affected your life.
I try not, and I think Dr. Schwartz's patient, and I really could relate and ignore it. And I don't know that that has always been the best thing, but I think coming to the NIH has been extremely helpful. And I remember at one point I was frustrated that nobody can figure out what we have. And Dr. Colbert, I think, gave me the best advice as a patient. And I think being a physician sometimes makes it difficult because I want to know what I have. And part of my type A personality doesn't help that. But Dr. Colbert said to me, he said, give it time. Don't let anybody label you. We will figure it out. But if we label you, we might give you the wrong treatment. And it's okay not to label you. And I think that was the best advice anyone had given to me, because I've been more patient with it. And I know having the group at the NIH has been wonderful. You've been supportive, and like Dr. Schwartz's patient, having trust in everybody. And I think as a physician too, and especially in the ER, and I think when you see pediatricians, everybody is so worried about getting people out the door that we don't always take the time to listen to our patients. And I think that's what has been made the difference at the NIH. You guys have sat down and you've listened. And it's been good.
Great. Well, thank you very much for sharing your story. I'm sure more questions might come up in the session. So I just have a couple more slides, and then I will open for questions. Thank you very much, Holly. Our next steps are to do a GWAS for FAPA. So we're in the process of recruiting enough patients to do that. And we believe that doing that will give us the opportunity to really compare the genetic architecture of these 3 disorders on the Behçet spectrum, and then further understand how what factors may determine where a patient falls on the Behçet spectrum. As Holly mentioned, diagnosing FAPA can be difficult. It's based on the clinical history. So understanding the mechanism may help us better develop objective diagnostic markers. And lastly, understanding the mechanism of these diseases, including diseases on the spectrum between existing classifications like Holly's, may open up new options for treatment, like apremilast, which has been approved for Behçet's disease, IL-12 and IL-23 blockade, or JAK-STAT inhibitors. In Holly's case, as she mentioned, IL-6 inhibition has been very helpful. So as we better understand the genetic architecture of these diseases, we can hone in on the right therapies for these patients. So with that, I'm really grateful to all of the clinicians and researchers who help us recruit patients for our genetic studies and are part of the FAPA Genetics Consortium. And I'm also very grateful to my mentors, Dr. Kastner, Dr. Remmers, and who are instrumental in this project, as well as all the other members of our team. Sorry, I don't have time to go through everyone's name. And also to all of our collaborators and patients and their families who donated their time and observations and samples. And I just wanted to thank Holly again for being here and for sharing all of her very insightful observations. So at this point, we're happy to take questions.
Okay, well, first of all, I want to, on behalf of all of us who have been listening, to thank the three speakers, and in particular the two patients who so eloquently described the difficulties of their disease. Several years ago, there was a postdoctoral fellow, a PhD fellow, who wrote to me and praised the idea of having the ability to see patients, which normally PhD scientists don't do. And he said that the virtue was that it puts a human face on a disease. And I've always remembered that, and I think it's a very powerful aspect of this course and of the basic principle. Now, we've had a series of questions, and so I will pose them, and then perhaps whichever of you feel is the appropriate one to answer, do so. So there was one question to ask whether there was a role for proteasome inhibitors, particularly targeting NF-kappa B, and in the treatment of autoinflammatory diseases, perhaps in general, but specifically some of the earlier ones that were discussed. So would one of you like to respond to that?
Well, maybe Dr. Schwartz, because she's been talking about NF-kappa B, would want to start off anyway.
Yeah, so I can talk about, at least in the setting of the HA20 and maybe some of the proteasome-related diseases. And in the setting of HA20 and in the diseases with an interferon signature, one of the things that we see is a defect in proteasome function. And because of that, you can get activation of something called the unfolded protein response that leads to enhanced inflammation and increased interferon signaling. And in some way, it's actually defective proteasome function and the buildup of these degradation products and that causes that inflammatory pathway. So in that particular scenario where you have increased type 1 interferon signaling, I think one would have to be cautious in using a proteasome inhibitor as opposed to some of the other targeted therapies that I had mentioned. I'm going to then pass it on to Dr. Kastner to mention whether in some of the other newer syndromes like Behçet that has been discussed, because there is that overlap then with the myelodysplastic syndromes.
Yes, thank you very much, Danny. And I totally agree that there are the proteasome related autoinflammatory diseases where, in fact, proteasome dysfunction can lead to inflammation. Now, in the case of the disorder Behçet, which is a disease that's caused by somatic mutations, acquired mutations in a pathway that leads to ubiquitination, and therefore, actually, the mutations impair ubiquitination and can lead to the accumulation of proteins that ought to be gotten rid of. There has been some discussion of proteasome inhibitors, but so far, to my knowledge, there have been no trials. And again, there is the caution that we're not sure that necessarily is going to be beneficial or could be harmful, but it may be that it would get rid of the cells that have the mutations. So that's at least the thought that maybe that would work. So right now, I don't think there have been any studies of proteasome inhibitors that have actually been conducted.
So we have a very important question, which is, are you still enrolling patients in these studies? And if yes, could you please point out some key eligibility factors? In other words, what do you look for a person to be recruited into these studies? Because many people have fevers, many people have some ulcers. Where do you draw the line? And if not, the person who asked the question wants to know whether you provide consultation and what's the best way of contacting you folks.
Well, I'll start out maybe, but I think my colleagues have also things that they would want to say. So first of all, we do accept patients into our protocol, our natural history protocol on autoinflammatory diseases. And we do also provide consultation to physicians who have questions for us. So with regard to the research protocol, the natural history protocol, clearly there are lots and lots of people who have undiagnosed fevers, that's for sure. And unfortunately, more of them than what we could reasonably accommodate. And so we are interested in individuals that may have unusual manifestations of their condition or may have totally undiagnosed conditions where there's the possibility that we might discover a new disease in the course of evaluating those individuals. And so we do focus on perhaps those very difficult cases or undiagnosed cases, although, you know, we will sometimes, depending on the circumstances, at least for maybe a single visit, evaluate someone who has a known disease with just maybe some problem that we could help with. With regard to the consultation, for sure, it's best if we receive that from the patient's physician, just so that we can get medical records associated with that. Certainly, I'm more than happy to respond to emails from people, and in fact, I do from both patients and physicians. My email address is dan.kastner@nih.gov. And well, I'm sure I'll have 100 emails tonight, but I'm happy to try to answer them anyway. And Kalpana and Danny, do you want to say anything as well?
I just echo that I am always happy to answer emails. My email is also daniela.schwartz@nih.gov. I personally am not recruiting patients because, as I'm not sure if Dr. Arias mentioned, I will be starting a tenure track position at the University of Pittsburgh over the summer, and will very hopefully be recruiting patients there, but no longer to the NIH Clinical Center anymore. However, I am always happy to consult or speak with any interested physicians or patients who have questions, although again, formal consultations should ideally come from physicians if possible.
To add on, I'm currently recruiting patients with PFAPA syndrome for the GWAS, so I need to get 1,000 patients, so it's quite a few. And in addition to that, I'm also seeing patients with symptoms on the Behçet spectrum, like Holly. I think she and actually her family, she mentioned her son also having other inflammatory features. I think families like her have really helped open our eyes to the connections between these disorders, and have made us think outside the box, and not just to say this is what you have, but to try to have an open mind and thinking about them. So I think it's really critical that we hear their stories and think about what the pathogenesis of their diseases are. And also, please feel free to email me. Same, my email address is kalpana.manthiram@nih.gov, and I'm happy to look into your or whoever is interested clinical features to see if they'd be eligible for my studies.
I think that, I mean, there's also a clinicaltrials.gov, which covers many clinical trials, which may be another entry. So one question that comes up, many of these disorders have the description of periodic. What makes them periodic?
This is
Well, that's definitely Dr. Manthiram's question, at least for starters.
I guess I'm studying the most periodic of the periodic fevers. So I think the short answer, we don't know quite yet, but we're beginning to look at the immune state during flares and non-flare periods. And we see this sort of oscillation that during the flare, there's high immune activation, but during a flare, there's almost sort of immune suppression, that the monocytes aren't sort of failed to activate normally. So I think that may play a role. It may be the natural way the immune system stops an overactivated state, but there may also be feedback mechanisms that lead to cycling, that lead to the extreme periodicity that is seen in FAPA, but there does appear to be these oscillating activation and sort of suppressed states during the flare and non-flare periods that we're looking more closely to define and understand.
So I guess as part of that is you measure inflammatory cytokines, for example, at one time, at many times in an individual. Is there a circadian rhythm? Is it related to other endocrine changes, menstruation? Is it related to stress? What about the second and third dimension of the changes observed in blood levels or even gene expression, other than at single times in given groups of individuals?
A great question. So I think with these periodic syndromes, it's really important to look longitudinally at patients and not just at one time period. So we've been looking flare and non-flare, but I think you're right that even looking more closely at more frequently, I think, at what happens as the flare begins may be critical, because by the time you're getting a flare sample, maybe the horse is out of the barn per se, but understanding what happens as an episode starts may be really important. And that's something that we're looking into doing in the future. As far as triggers, I do have some patients who find that their episodes are worse. These are patients who continue to have flares when they're older, that their episodes are worse with stress and with females with their menstrual cycles. I was wondering, maybe Holly could chime in about triggers for her flares and what things have made it worse for her.
I think my stress definitely is a trigger. I worked night shift in the ER for 6 years continuous when my kids were smaller. And I think that's when I really started getting worse. I think the lack of sleep, the interrupted sleep. Now that I'm not working in the ER and I'm more on an 8 to 5 schedule, I think that has helped. Our son, he's now in Alabama going to college where it's warmer. I think we're in the Northeast. I think now that he's in college, he's getting less flares. So I tend to think maybe the cold sometimes aggravated his, because he was always worse in the late fall, winter periods.
You brought up a timely question, which comes up in every one of these sessions we have, and that is, what's the effect of the COVID infection and pandemic on any of these diseases? Do we have a handle? Does the virus itself participate in some of these inflammatory processes? What do we know about the relationship between COVID and the autoinflammatory diseases, both as clinical manifestations and also in terms of the underlying science?
So I actually want to point out that one of our colleagues, Dr. Rafaela Goldbach-Mansky, published a paper in the Journal of Clinical Immunology that was released this week addressing that exact question. And I read it this morning in anticipation of this exact talk. And she followed several patients with mutations in NLRP3 and in proteasome genes. And those patients, some of them did develop COVID-19 infection. One of them was severely affected. That patient had underlying lung disease and responded well to the immunomodulators that are used for severe COVID. That patient received an IL-6 inhibitor. The other patients had mild disease, and many of them had COVID vaccinations that were tolerated really well. A couple of them had to take a few extra doses of anakinra. And so I would say that based on the best data we have, which are those data, and observational sort of, I guess, follow-ups of our patients in the cohort, that the vaccination is well tolerated in our patients. Some of them do need a couple of extra doses of their immune modulators, and that similar to many patients with rheumatic diseases, there is a probably increased risk of severe COVID infection. They are patients with chronic inflammatory diseases, but the majority of them can do well with appropriate monitoring and care. Although it's good to involve their providers sooner rather than later. Dr. Kastner or Dr. Manthiram, do you have anything to add from observations of your patients?
Yeah, I would say that I totally agree with what Dr. Schwartz has said. And I would say that on balance, we definitely recommend in favor of vaccination and in favor of immunization against SARS-CoV-2 in patients with autoinflammatory disease. The risk-benefit ratio is definitely in favor of having vaccination. I can tell you that occasionally there are some anecdotal cases that we've seen. I saw one patient a few months ago, a woman with familial Mediterranean fever who had been in remission. She was in her 60s and had been in remission for a number of years and had her vaccination, and then she had some attacks of FMF that were sort of stirred up by the vaccination. But still, we were able to get that under good control easily, and certainly the risk associated with COVID is substantial, and so we do recommend in favor of vaccination for our patients.
And also in my HOT20 patients, the few that have had some increased symptoms of their disease with the vaccination, it's been very manageable, very easy to control, and we've been very happy to see that they tolerate it well, and it's on very much recommended for that group.
With the patients with FAPA, I think the concern was that with the heightened myeloid cell and T cell function, whether these patients might have a hyper immune response to the virus, but we haven't seen that to be the case. We have most patients respond or don't seem to have severe side effects with the vaccination. I think with since Holly's here, her son did have a flare, I believe, after after the second dose of the vaccine, and we have seen that, as Dr. Schwartz and Dr. Kastner mentioned, but was able to be under control.
It's an interesting question, Dan, that maybe you would refer to. Given the geographic history of FMF, does it have any bearing on the origin of the Ashkenazi?
Well, it's a very interesting question with regard to the relationship between Fmf mutations and the Ashkenazi population. And certainly we do see, as I was showing on that map, a particular mutation, the V726A mutation, that is seen in the Ashkenazi population and is seen on a particular haplotype, that gives us reason to believe that in fact, at least the origin of that population for sure is in the Middle East. And it's incredible. When we went to Israel back in 1989, when I was in Israel drawing blood on all of the patients, I went to a couple of Druze villages. So the Druze are this population of people that live in isolated mountain villages. They have their own religion and have stayed isolated from the rest of the population of the Middle East for the last 1,000 years. And they have the same mutation on the same haplotype as the Ashkenazi population does. So it's really fascinating, thinking about, what this tells us in terms of the history of these people. Of course, it can't tell us exactly what's happened step by step as they have spread across the world, but it certainly does tell us something about their origins.
Well, it's what Chaim Sheba, for whom the Sheba Medical Center was named, referred to this as the genetics of migrant populations and studied them. So is there any information, for example, about those individuals, say from North Africa, South Africa, I've forgotten some of the names, where there were claims of actually being descended from ancient times, from the Hebrew group, and some of them
Are you talking about the Ethiopian?
Have some evidence for it. Does it? Yes, I'm sorry.
Are you talking about the Ethiopian populations that immigrated to Israel in the 1980s and 90s?
Yeah, that's part of it. And then there's I forgot the name of it, the tribe in South Africa that's very primitive, which also have the Middle Eastern gene marker on the Y chromosome.
Yes. So we haven't looked at FMF mutations in those individuals, but you're right. I mean, it's a fascinating thing, and the anthropology of it all. I mean, that's part of the reason why I love doing this, is just that, having a chance to look at those kinds of things, and it's people, and that's the most wonderful thing of it all, is just the fact that we're dealing with other people.
So one last question, of a practical one that has come up before. You say that the patients whom you study are referred by their physicians to you. But what about the individual patient, say in a family, where, a child, lots of children have recurrent fevers, and there aren't many answers. At what point do you draw the line as to when a physician should begin to think of these diseases, the clinical scope of which seems to be expanding, not contracting? So what would you advise both patient and physicians as to when to begin to think about?
Well, it's a great question. And for sure, at least in terms of thinking about genetic disorders, someone who has a very early onset and who has severe disease, we for sure think about the possibility of genetic disease in those individuals. Nowadays, with the availability of genetic testing, it has become easier and easier, and physicians are doing it more and more just to screen for variants in the periodic fever genes in panels that are available from some of the commercial genetic testing companies. And so in some cases, that's the way it happens. But I think that, your point that the scope is increasing and that the numbers are increasing. And so it's important for physicians to think of these diseases sooner rather than later. And certainly I would rather be asked a few too many times and then find someone who does have one of these conditions that are treatable. And in some cases, like for familial Mediterranean fever, at least some people that have that disease can develop amyloidosis, which, as I mentioned, can lead to things like kidney failure. And so I'd much rather err on the side of asking too many questions than not asking enough.
Yeah, and I just want to point out that sequencing is, as Dr. Kastner mentioned, becoming cheaper and cheaper to the point where many patients can even go and get an exome sequence done privately. And so it has become easier to say this patient doesn't fit into a bin of a certain disease. And maybe looking back in retrospect, they have had oral ulcers or genital ulcers. We've had a patient who came to us at the age of 45, and oh, by the way, he's had oral ulcers since he was 8. And no one thought anything of it because everyone gets oral ulcers, don't they? But not everyone does. And that patient had a haploinsufficiency of A20. And so it's not it's much more available to just get that genetic test, and worst case scenario, you'll get a variant of unknown significance or nothing, but maybe you'll find something.
So one last question, Daniela, maybe you can respond to this. Why does the disease seem to go away in some patients with time, or at least greatly become modified? Do you have any thoughts about that?
Are you talking about haploinsufficiency of H1 going away over time?
Well, yeah, my understanding from some of the presentation is that it doesn't always get worse. And some of these.
That's actually for Dr. Manthiram. That's her disease, not mine. Mine, unfortunately, I wish very much that it went away, but unfortunately, no, mine does not. I think that's more Dr. Manthiram's disease.
I guess for most patients with FAPA, the episodes seem to resolve, or at least the fever part resolves. I think most patients continue to have the aphthous ulcers afterwards, resolves around their before they reach adolescence. So I don't know exactly why, but one reason may be involution of the tonsils. So as I mentioned, tonsillectomy leads to resolution of the fever episodes in most patients with the disease. I mean, Holly is sort of an exception, but I think the tonsils may naturally get smaller with time. So that might be one factor that plays a role, but we haven't fully understood, especially because these genetic risk variants that we identify are still present even beyond childhood. So I think, more work is needed. And just to your prior question about when to suspect a periodic fever, I just wanted to add that, normal children can have very frequent viral infections, over 10 infections per year. That can be normal. But I think one question that really is important to me is, are the episodes all the same? Like, do they, and these parents will I just know that's what it is. And I really trust them because they sort of get a sense of the pattern of the disease. So I think that stereotypical nature is really important. And I think if anyone suspects that their child or themselves has a periodic fever, a really helpful thing is to start maintaining a symptom diary or fever diary. So we can start seeing if there's a pattern to how the episodes present.
All right, well, all good things come to an end. I think we thank you all for this really exciting Pandora's box that you opened for those of us who don't think about this all the time to think about. And I trust that that's true for the large audience that will be watching. In particular, thanks to the two patients who kindly shared their personal experiences, which is such an important item. I just would mention in closing to remind you that next week we have an extraordinary bridge, the bridge back to the creation of life. And so we will have a leading biochemical, evolutionary biochemist, Nick Lane from University College London, and a leading cell biologist, Jennifer Lippincott-Schwartz, who are going to discuss the origin of life as viewed through their perspectives of their individual research. So thank you all again, and please enjoy. Thank you.
Thank you.
We're off.